Okay.
We can
There
carve
you
that out.
go. All right. So thank you, everyone. So we're going to talk about polygenic scores today. Focus on polygenic risk scores, although any sort of polygenic score will work in the same way. And just to sort of level set everyone of what we're talking about. So on the left, we're looking at more Mendelian conditions, so cystic fibrosis, where variants at a single locus have high sort of impact, high odds that and lead to sort of more Mendelian presentation of disease, as opposed to when we think about more common complex disease, where multiple sites, many sites throughout the genome, all contribute in a small way to the overall phenotype, and trying to tease out the sort of more common complex things to understand people's risks for developing these more common conditions. The next slide. So again, looking at things at sort of this more penetrance level, we're looking at things a little bit differently. But on the left-hand side, that sort of light tan bucket, the GWAS one in that bottom right-hand corner, these are typically the variants that are going to go into a polygenic risk score, which are the ones that are sort of identified by GWAS studies that have really low sort of overall odds ratios, but collectively have an appreciable odds. And more, what's happening is that sort of reddish bucket in the middle. Those types of variants are now being incorporated into the scores too, where you have these low penetrant or more risk allele type variants that have sort of strong effect sizes on their own, and they end up being incorporated more into the model. So a common one might be APOL1 in chronic kidney disease. So there's also different things that are happening there, but that's sort of where the field's going, is we're getting more power for these sort of low penetrant risk allele type variants, and that are things that are a little bit more rare. We can go to the next slide. So what happens in a polygenic risk score is you have this sort of GWAS data, and we're going to show this a little bit on the next slide, I think. And then you sort of sum up the weights across all the different sites in the genome, and you typically will see this sort of standard bell curve. Um, some of them that have really high impact variants don't necessarily follow this typical bell curve, but most of them follow this standard bell curve, where then you have the individuals in the far right-hand side that have the highest sort of values for their polygenic score, and that's equatable to sort of the highest risk. And that's one of the things that you oftentimes study is that, okay, are the people in that high bucket actually having the highest risk compared to everyone else or compared to the average person in the group? And that's sort of what we're looking for, is trying to tease out the people with the highest risk here. Go to the next slide. Um, and then, yeah, just another form of risk modeling that sort of clinicians are used to. We're trying to stratify people using polygenic score. And the reason we're doing this is to think about how people may want to be, or how clinicians may want to treat patients slightly differently, and sort of which pathway they might go down. And so if you have a good discriminative model like you do here in Model B, you can find the people that are high risk, and they'll have different sort of treatment pathways than people in the other buckets. But you really need to make sure that the model has this sort of discriminatory power to be able to do this. Or you can go to the next one too. And as I said before, we're focusing on risk scores, but more and more polygenic scores are being used for lots of different things. And so risk prediction is what we're talking about. That's sort of being used since birth. Oh, sorry, not it's you can generate these scores sort of from an early age, and sort of because your germline applies sort of going forward. But more and more, there's other things that are happening here. So there's a lot of work being done to support polygenic scores and sort of a differential diagnosis. So which pathway might a clinician go down if it's an atypical disease presentation, and there could be a couple different ones. There's some things now in prognosis that are coming out in sort of different levels of treatment. So this is again an emerging field, most of which has started sort of on this risk prediction, but now there's different things. Different stages of disease, and oftentimes like secondary hits or something like that. So progression is another one where it's trying to understand if someone has a disease, what's their likelihood to progress to more severe disease, that sort of thing. And so there's a lot more different contexts that people are now developing these models for, in addition to just sort of the standard risk prediction. We can go to the next slide. And this is what I was talking about sort of in the model development, where you have your GWAS data, and there's lots of different algorithms that are out there for sort of developing your polygenic risk score from the GWAS data. But you typically then have a sort of a training set and a validation set. You train the model in the training set using uh, the variants that are in the GWAS studies with their sort of effect sizes, plus any other sort of demographic type information that might be important for the model. And then there's different ways to sort of evaluate the model in the test set. We're looking at a few down there, but typically you just want to show that you have a increased odds ratio for the people with the highest risk score. You can look at sort of the odds ratio per standard deviation, that sort of thing, and really being able to tease out that the people with the highest values really do have the highest risk odds here, and vice versa for the lowest values. All right. And then this is probably just one last thing to get into. In terms of sort of thinking about polygenic scores, there's lots of different models out there, lots of different ways to do it. I tend to think of things bucketed in sort of two things. Some of them are just using only those variants that meet the genome-wide significance level within the GWAS study. And so typically these PRSs are in the 300, or sorry, hundreds to thousands of range, as opposed to some of the other models that take in every single variant that was identified within the GWAS, whether or not they have large or small effect sizes, and sort of then generate the polygenic risk score off of those summary statistics. And so there's just a few different ways to think about that, whether you have, again, maybe hundreds or thousands of variants in the model, or even in the tens of variants in the model to sort of millions of variants in the model. Not that one works better than the other, but there's just different ways to think about applying them. All right, I think that's you can skip this. I guess the only other thing that's important to think about, we can go to the next slide, Shruti. Um, there is sort of some challenges still in the field with portability between ancestries. And so just because a lot of our historic data has been generated on people with European ancestry, that's where most of the models have been trained. This has gotten much better. And if you go to the next slide, there's new models too for how we can sort of mitigate some of these biases. So there's a lot more new models that are basically trans ancestry. And so you're able to sort of use multiple different ancestries within the model training itself. And as we get more and more of these large data sets online that are sort of non-European some, genetic similarity group, we have the ability to really train these models that not only then perform better in the less represented genetic similarity groups, but also in sort of the Europeans as well. But if you go to the next advance that, there still are some differences. This is hard to see, and I apologize for that. But if you're looking at the thing on the graph on the right, the figure on the right, the red bar indicates the people in the highest risk category, and that is for different. Each of those four figures in there are for different genetic similarity groups, and you can see there still is different sort of discriminatory power. Partly that is again due to the maybe smaller sample sizes. Partly it's due to maybe different linkage disequilibrium patterns within the different similarity groups. And so just because we still need a little bit more power, or there still might be some biological reasons, the PRSs aren't always sort of apples to apples in terms of their odds ratios, even if we are able to discriminate people from the high risk bucket. And that's just an important limitation that still exists and one that we want to just be aware of. All right, you can sort of keep going. Um, yeah, I think that's probably good, truthfully. If you want to, we have a lot more stuff thinking about. Actually, if you go to the ancestry adjustment, because we do talk about that a little bit. Um, sorry to skip all ahead. Yeah, so let's go back one, I think. So one of the things, and this is really hard to see, and I apologize, this figure needs to be improved. One of the things we still see, even when we have sort of these trans ancestry scores, is that we still have different raw distribution patterns between genetic similarity groups. If you look down in the bottom right, the type 2 diabetes one is probably the best example. And I know it's very hard to see, and I apologize, but the black line is basically the distribution for people with European ancestry. And you can sort of see the two bars, like a reddish one and a greenish dotted line to the right, that are for different genetic similarity groups within our biobank. And you see that there's actually a skewing of the raw scores. And so one of the things that we've implemented, a lot of people have implemented as well, is sort of to get everything sort of aligned, we end up creating a model where we remove any of the PRS that's based upon the genetic similarity group. So we have this basically PC adjustment step that Shruti will show. And we get everyone sort of in the same line, the same curves to be able to have a little bit more, I guess, especially when we're thinking about this, sort of launching it sort of a single score for the metric for determining who's at high risk or not at high risk, or if people are sort of admixed and we're not sure what bucket they belong to. It gets everyone sort of on the same plane, so we can sort of apply that standard threshold more easily. All right, I think that's probably a good background if we want to sort of get into the model. And so what Shruti is going to show is the this whittle that we've created for basically running polygenic risk scores once you have a known score. And actually, if you go back a couple slides, Shruti, to the PRS mix stuff, I can describe that a little bit as well. So one of the things that we're doing in this thing, we have the three different steps within our PRS sort of whittles. The first one is just getting all the inputs on the same page, basically. So it's just standardizing all the inputs that go into it. One of them is training the model, and one of them is actually doing. So one's training the ancestry adjustment model, then one of them's generating the scores. But one thing we've done back a couple slides, I apologize. Keep going till you see the one more. Yep. All right. So one of the things that we're doing is also incorporating this method called PRS mix, which is sort of an ensemble method. And any sort of ensemble method would do, it doesn't need to be PRS mix. But basically, what we people have shown is that while each individual PRS sort of works well, there's oftentimes not good correlation between them. So here we're looking at a couple different scores for CAD and type 2 diabetes. And what we're showing is that if we're looking at all the different polygenic scores in the PGS catalog, and we're looking at people in the top 10%, there actually isn't a lot of good overlap between them. And so what our collaborators have shown that we've implemented to this, if we use this PRS mix method, I think if you go to the next slide, it shows a little better. Um, we have the ability to now get a lot more robustness for who is in that top 10%. So basically, if you now use, you weight each of the individual PRS scores and you determine who, you weight each of the individual PRS scores. And so then someone has a overall polygenic risk score using this PRS mix method based upon all the individual weights from the individual scores. And there's then much more stability in who's in that top 10% whenever you add a new score. And typically, we don't have the slide in this presentation, but I think when you get to like 10 or 15 scores in the catalog, there's oftentimes really good stability within sort of adding any new scores. And so it really has shown a way to sort of have much more confidence that if we're putting someone in the high risk category, they're going to stay in the high risk category. Regardless of sort of which new scores come out. And so that's something that we've actually built into the Whittle is the ability to use either a single score or this sort of ensemble type method within our Whittle. So, all right, now you can go ahead, Shruti. Sorry.
Okay. Thanks, man. That was really helpful. So diving into running this in AnVIL. So like Matt said, there are three steps. So just to keep things clean and to let people customize the whittles that we have, we've actually created three smaller whittles that you can run. So the first one is an input preparation. So considering that uh, any input files that you might want to run this analysis on might not be in the exact format that the Whittle is expecting. What this input preparation is going to do is going to normalize all the chromosome names, the kind of variants that are included from the reference file that you would use, the query VCF that you want to run PRS on, or the PCA variants that you will want to use. So I would recommend that running in the input preparation is really important for future success of the scoring pipeline. So that's the first step. And the second step would mostly be creating the model for the particular disease condition that you're looking at. So it would take in most of the inputs that you use in like the first step produced. That would go into the second step for the model generation. And what running that Whittle will give you is a JSON format file that will have all the like the disease condition, the various manifest files, and like everything that will be needed for your PRS scoring Whittle, which is the third step. And what this Whittle is going to do is it's going to create like raw scores, like Matt mentioned earlier. So that's the PRS raw score, which will then get adjusted using the ancestry adjustment model you created in step two, and give you a set of adjusted scores that are like through the ancestry that you have like in your reference VCF that you provide. So this is kind of like the three-part whittle that we have to run PRS on Anvil. Going into it a little bit deeper. So say, for example, you have 10 VCF files that you want to run PRS on. You don't need to run the step two, which is the model generation step for every VCF. You can run that once. So that'll give you a model that you can use for all the rest of the VCFs that you have. So this is kind of a work, like a flowchart that shows you what you could do if you have like an ancestry adjustment model already. So basically, you have four primary inputs: your query VCF, which is the VCF that you want to run PRS on; a weights file, which is the PRS weights for that particular disease condition; a PCA variance, which will be used for PCA analysis; and the model that you create in step two. So the whittle will first score the query VCF. Then that will give you a raw score. And that raw score then gets adjusted to give you the adjusted score. So this is a very high level overview of what the whittle is going to do. Moving on, this is a little more in-depth look at, and again, this is really small, but I added this just to highlight the fact that there are a lot of things going on inside the whittle. And if there's a scenario where you want to run a mixed score, like Matt described earlier, it's basically a scenario in which you have, for a particular disease condition, multiple weight files, and each of the weight files has like a weight itself to it. Like, for example, if you have A, B, and C, three weight files for a condition, each A, B, and C will have a particular weight, and then that weight is going to get used to create a weighted average score. So that is going to be your PRS mix score. So like here in the blue part, it's creating the raw score. So basically, if you have three weight files, it's going to create three raw scores for that one VCF. And then the orange part is going to create a weighted average. Basically, use the three scores, apply the weights to it, and give you a PRS mix score, and then do the PCA and adjustment on it, which gives you the PRS final adjusted score. So that's the score that you can use for earlier visualization and just looking at what kind of trends that this VCF is showing. This whittle is flexible enough to do a single score PRS analysis as well as a mixed score. The only difference is that in a single analysis, like the name suggests, there will be only one PRS file that you're using. For a mix, there'll be multiple, and there'll be an additional file input that you would provide, which would be the weights for each of the PRS mix files. So um, Next, okay, so even when you decide to, for precursor, like the workflow, the workspace that I'm going to point you to already has a lot of demo data that you can use to run the whittles just to get your hands dirty and see what is what. But moving on, like if you want to use your own data, there are some requirements that the whittle is expecting that this data will have. So firstly, the query VCF that I was talking about earlier can be single sample or joint sample. That's not a problem. Both will work the same. Just a heads up, if it's a joint sample and it's a relatively bigger VCF, you might have to add like a little more memory, and that's an option in the workspace, which I will show you in a bit. And secondly, if you're trying to use your own PCA variants, I would just recommend that you check if the PCA variants are present in both the reference VCF and the query VCF that you're using. Otherwise, things might break. So just a quick heads up on that. And the weights file that you use. So I usually get my weights file from the PGS catalog, and most of the files are in the format that I want it to be. But in case the file that you're getting is not following the same format, what the whittle is expecting is that there will be three columns. The first one would be the variant ID. Like you see here, it'll be the chromosome number, the position, and the effect allele and the ref allele. The second column would be just the effect allele, and the third column will be the effect weight. As long as you have these requirements, it's a safe bet that you can use your own data on these whittles. Um, Okay, so moving on to actually running it. So thanks to the Terra team, our workspace is now public. So if you go to Anvil and go to the Workspaces tab and search for under Public Anvil Polygenic Risk Course, you should be able to find this workspace that we have set up for you. And using the three dots, you can clone the workspace, name it whatever you want, add a billing project, or I think it's called billing project. So basically, whatever workflows you run will be built to that project. And I would recommend to start, you could use the demo files that we have. And this is a request or paste bucket. So whenever you want to copy the files into your cloned workspace, I would recommend this command, Google Cloud's G-Cloud Storage copy, add your billing project. The source path would be the path of this public repo, and the target path will be your path. And all these commands are already present in the dashboard view of the workspace that I'll show you in a bit. So no need to like copy that down right now. You have access to that already. Um, Next, so say you don't want to clone this workspace, you have another option if you want to run these vittles in your already existing workspace, and that would be to import it from Dockstore. And since this is not the easiest thing to show on a shared presentation, I will show you how to do that on my computer. But you can do that from Dockstore. It's also publicly available, and you just need to export it and add it to your workspace of choice. And from here, I think it's a good segue for me to shift to my browser and show you how to actually run things. Okay, so here. So what I was talking about, so if you go to like this is probably where you would log in, and this is what you would see. So the first thing you would go is here is the public workspaces, and I don't need to really search because it's right at the top. But if you want to, you can search there. Just type in Anvil PRS, and it should show up. Opening that will take you to a dashboard, and this dashboard is going to talk about all the like what we're trying to do in this workspace, what the contents are, and the three steps that you can follow. There are more than three, there are five steps. The first one is cloning it, and the second, here is the command that you can use to copy the data. Um, Okay, and one other thing that I would like to mention, I mentioned that there's a project ID and the bucket name that you need to use. If you want to copy the data, you can find that here. So under cloud information, your Google project ID is the ID that you would use for your billing project. And this is the bucket ID, which is basically the target location of where you want the data to go. Moving on to projects, the middle stuff, you can go to the workflows tab here, and the three steps that I was describing earlier are here. Um, but before I dive into this, I do want to show you how to get it from Dockstore, just so you have like another way to do things. So if you go to Dockstore and if you just look at like search for Anvil. Probably shouldn't have searched for something more. Okay, there you go. So you can see the three whittles that I was talking about earlier are here. So if you go to any of these, say this is the third step, which came up first, but what you would do from here is go to launch with on the right, go to Terra, and select, it might ask you to sign in again. Select the workspace that you want to go to. Just select anything that is already existing. Um, let me see if I can select something that. uh, Here, this is fine. And once you select that, just import it. It will take you to that workspace, and you can start running your riddle from there. That's how to import it from Dockstore. But let's look at running it. So I have already cloned this repo, and I've called it just demo, and you can call it whatever you want. And this will clone whatever is on the dashboard, some of the data tables that I will talk about in a bit, workspaces, workflows, the three riddles. And yeah, I think those are the three main things that will get copied over. The data files that I was talking about earlier, like on the public workspace, you'll find here, like browse workspace files. And that should have like all these things that I think would be a good place to start using this whittles from. So these 1 kg Bull38 is basically the 1,000 genome shards, or since it's a huge file, we've broken them down into shards, so it's easier to handle. So you can copy this. The HTTP samples are 10 query files that I'm planning to run PRS on. This is a sample weights file. You're welcome to use your own weights file, no worries. This is a demo plot file that you can use to visualize the data at the end. There's an input file requirement just in case you want to look at what you need to have for your inputs. A PCA variance file, I would recommend that you start with this file and then you can edit it as you need, and a renamed chromosomes file. So this is a very small kind of a dictionary for all the different forms of chromosome IDs that you can find in VCFs. So what this is going to be doing is just renaming all your input chromosome files, chromosome names in your input files. So it's like homogeneous and uniform, and does not have like, oh, variant not found kind of errors that can crop up. So feel free to copy these files to your own bucket, and you can use that to um, run your riddles. And I have, like I mentioned before, I already copied them here. So similarly, this is my cloned workspace, and whatever riddles I have, I can just directly run it from the locally available data. So diving into the first step, the PRS input prep. Um, there's one way to look at what we're actually doing, and once you like open up a whittle, if you scroll down a little bit, you'll see script here. So we have a lot of our tasks in this GitHub repo. You feel free to look at it if you want to dive a little deeper. But this workflow basically talks about everything that we're doing. Like we are renaming the weights files, the PCA variants, um, and then like getting like doing a bunch of weight calculations. So Terra VM gets the exact amount of disk space that it needs, doesn't break that way. And it uses a helper task like from those links above, and does a whole bunch of renaming and cleaning up the data so that whatever outputs you get from this whittle are ready to use in the next upcoming whittles. So that's this whittle if you want to look in a little more detail. Um, so first, there are two ways to run this whittle. One is to directly give it the files that you already have copied into your workspace, and the other is using a data table. But since this one, we don't want to run like 10 input prep whittles, because this is fair warning is going to take a lot of time, because the reference VCF that you're using is huge. So be prepared that this is going to run for a few hours at the least. So you don't want to have like 10 tasks that are running for like 6 hours. So what I've done here is like instead of running it for one query VCF each, I have the option of running it like a array of files. So you say if you have like 10, like in my case, if I have 10, I can list it 10 here, 10, all the 10 samples here, and that will work fine. But right now, I have just put two just so that it runs pretty fast. There's the PCA variants here. Um, so the first time you clone your workspace, I have like template or placeholder paths that you can that'll help you decide what files go where. So like for example, here you just need to click this browse buckets and it'll let you choose this PCA variance file. Similarly, well, you cannot do this for here because it's a array of files. You will just have to copy the GS links or the Google storage links and place them here inside strings separated by commas. And for the stores, this is basically the path to your reference VCF shard. So here for me, it's 1 kg. So I can just place the Google bucket here, and that'll be it. And the next input that you would like to give is this is the path to your target, wherever you want the cleaned up reference to go, so you don't have to, keep doing the reference renaming. And similarly, so like we talked about earlier, this whittle has the capacity to PRS as well as PRS mix. So that's why this is an array. You can provide one or more PRS weights files here, and all scenarios will work. So again, in double quotes, separate by commas if there are more than one. And one other extra input is this renaming chromosomes lookup. So I do have like a no rename flag here that you can put to false if you put to true, sorry, if you are 100% sure all your VCFs are in perfect condition, there's no renaming required, and they're good to go. You can say true, like don't rename, but I would advise against it just because it will break in unexpected ways, and you don't want to spend time just like 4 hours to get to a point where it's like, oh no, this is not found. So I would suggest just do a rename for the first few runs, and then once you get more comfortable, you can skip it. So basically, if you leave this blank, you need to provide a chromosome file, basically a lookup for the chromosomes to be renamed. And that's that for this whittle. Um, to save time, I already ran it a bunch of times. I also have a failed sample just to show what it looks like if it fails. But here, but once you add all your inputs here, sorry, I'm going back, but if you add all your inputs here, hit launch, you can add a comment just to describe this run and click launch. It will take you to your submission history page. This is how it will look when it's running. This will go from like launch to submitted to running and succeeded or failed. But this is your submission page. Uh, from here, what I would do to look at what's actually happening is click on this job manager. This job manager is going to and I apologize if I'm giving a lot of Terra Anvil background, but I think this helps in understanding what's happening. Um, so you can see the different tasks that are running in this whittle. And these are in this column is all the inputs. So if you click on it, you can see what inputs going into which task. And similarly, what outputs are coming out of which task. This is important because these are the outputs that you're going to use in the next whittles. So, like you see, a lot of it got cached. Like I run it a bunch of times, so it's using the older data, which is something you can do too. Like if you're running something multiple times and you know that you've already run this earlier, you can enable call caching. Like here, call caching is enabled, and that'll save you a lot of time. I highly recommend that. But like you see here, like this, for example, which is basically trying to work with the reference VCF took like 4 hours. So like I said, it's going to take time. Be prepared. The first step is very time consuming. Um, yeah, that's for that. I'll go into debugging a little later. Let me talk about the PRS model as well. So once you have all your inputs from the step one, you would go to the model generation or PRS model step two whittle. Similarly, I'm going to still run it through file paths just to keep things clean. For now, like the base file that I had is for CAD. This is a string value. You can call it whatever you want, however descriptive you want it. This PCA variance again is the clean variance from step one. So you will notice that it's going from a PRS input prep. So the first whittle created this subfolder, and under the task, relevant task, it created a renamed PCA variance. Whether or not the file got renamed or not, this file will definitely get created if you run the input prep whittle. But if you're, like I said earlier, if you are sure that your PCA variance is perfect, go for your original file. But fair warning, it might throw errors later. Um, Similar to the PCA variance, the query file also will come from the input prep whittle. And in this case, to generate a model, we're working under the assumption that all the query files are like from a similar source and have similar set of variants in them. So what I'm doing here is instead of giving it like a huge list of files, I'm just giving it one file, and that file is more than enough to create a model that you can use on the other files. So I've used one of the GDP samples here. And I'm also using like again from the input preps whittle, the reference VCF. That's a clean 1000 genome reference VCF here. And again, from the input prep whittle, the cleaned weights file. Again, this is an array because you do have the option of providing multiple sample files, multiple weights files, I'm sorry, and that will work just fine. Similar to the first one, again, you will provide the same renamed chromosomes, which you can select from here. And that's it. That's for the model files. You hit launch and it'll take you to the submission page, where again, you can use the job manager to look at what's happening. So this is the primary task that is running. You can see a whole bunch of subtasks inside it, and the bundle adjustment model, or the last whittle, basically is going to give you the output. Since this whittle is going to give you one manifest JSON, I would recommend just going to the outputs file and getting it from here. So this is the CAD is the string that you gave at the beginning. So CAD model manifest is the adjustment model that you want to use for your scoring later on. Um, it also gives you a population adjusted scores, TSV, which the scoring whittle doesn't use. But if you want to ever look at the population adjusted scores, this is where you can find it. That's step two. A major chunk of our work is now done. This PRS scoring, I swear, is the fastest way to do fastest part of this whole process. So here you would provide it. Here. So let me show you here because that might be easier for you to look at. Okay. So here there are just two primary inputs. There's the VCF file from step one, it's cleaned, and the model manifest again from step two. So the JSON file that we looked at earlier that goes here. And if you want to still do like a final renaming lookup, you can do that here. But I believe at this point, everything is cleaned enough that you don't need to do it. But in case you run into a scenario where things are not looking as they should, and you're running into errors, here's and you have the option to run this with a renaming lookup here as well. Also, if say you're working with like a larger PCA variance file, or you think the files that you're using are on the larger side, I would recommend increasing this memory. It's usually around 8 GB. I would bump it to maybe 16, just to give it a little more wiggle room. That's third whittle, and like I said, this is the way to do it with file paths. So the second way, the data table, is what I want to talk about next. So um okay, here, let's look at a data table, for example. When you clone the workspace, you will have this data table loaded by default. But this data table has all the samples from the original workspace, which you might not have access to. And what I would recommend is that I would like download this, like select it, export it, and the TSV that you get, just change the bucket names or the paths to the cleaned files from step one. and which is what I've done here. And this is all you need from here. If you look at this, for example, um, hopefully it shows me the path uh, here. So it's not great to look at, but if you see, it's like a submissions path. So it's from one of my, excuse me, one of my input prep runs, and that's a renamed query VCF. So that's copying to wherever you want it to copy. So and just create this data table. And what this will do is that say for example, like here I have just 10, but if you have like more than that, like you have 50 or 100, you don't want to create this submission for 50 or 100 samples because you're going to have to do the same thing again and again, just, changing the file path. What, uh, what this data table concept lets you do is that if you go to workflows with inputs defined by data table, select your data table, the renamed one, and here. So you can select as many as you want, as few as you want. And say, for example, just to show you, let me select all 10. And I say, okay, the only change here is that instead of providing the full path to your input VCF, what you would do is more like a object-oriented style. There's like this dot VCF file path, and that is this column name. So you do this cloud file path, and that will just take that particular file path for every run here, and the rest remains the same. Like the model manifest goes here. I've put this here just for safety, and I have added some extra memory just in case. Um, and that's all. You launch, write in a comment, run step 3, 110 HTTP. Okay, and hit launch. And what that would do is it's going to create 10 runs for you. You don't have to do it one at a time. Like I said, it's still launching. If you hit refresh, it probably would have submitted by now. Yeah, there you go. So once it submits it, you get all these nifty links to go to the job manager, which we saw earlier, a workflow dashboard, which gives you an overview of that task. And this is the directory in which all the files particular to that, like submission, are going to be saved. So let's go to the job manager, see what's happening. Um, it's going to run for a while, so while that runs, I can quickly show you the workflow dashboard. Again, you have a job manager, the directory, and the log files here. This is a good place to look at, what tasks are running, how they're going, and things like that. Similarly, the execution directory takes you to Google Cloud, and it lets you go inside. Like this is your primary bucket, submission ID, workflow, and your submission. Yeah, there's a workflow ID, I think. And inside that, this is the task that's running, and it gets divided by every workflow, every task inside that workflow, everything that it's doing. Like for example, like now it's taking it from the cache, but it'll give you outputs to whatever got renamed. So it's a long-winded way of finding your files. I find it easier to go to the job manager and just getting it from the output section. But if you want to look at all the extra files that created, or the log files, or just look at, oh, what did this task run? Like what's the script that is running on the backend? That's where you would go and find it. So having said all that, these are the 3 steps to run PRS. And let me go back to my presentation and I can show you what you should expect. Uh, okay, that's the right screen. Um, Once you have all your pair scores, the step 3 is completed successfully. This is like a snapshot of the final adjusted scores file. So these are all your PCs. So the first 2 that are most relevant, highest significance, and going to 10. And that after that, there's a score, an adjusted score, and the percentile. So this is not important, like this is not like very useful if it's just one sample, but to compare. But when you run like multiple samples, or if you have like a joint VCF, you would see per sample data scores, and then that's something you can use to like plot it. For like one of the plots that we created here is like the red is basically the population data, and this is just one sample that we ran, like say the one that we looked at earlier. That's the blue dot there. So you can see how it's trending compared to the reference that we used. Okay, so this is all I have to say for PRS, and a quick recap would be like look at the cloning workspaces, importing data, data tables, models, and PRS generation, setup and running whittles, tracking jobs. Oh, I do need to talk about a little bit of troubleshooting and the results. So here, let me go back. Um, so if I show you one of the like this field, this is kind of a silly reason why it failed, but I can show you how I knew what went wrong. So for example, this is the task that failed. This is pretty straightforward, but this is how you would look like. The errors is will sometimes are pretty descriptive. Like in this case, it's showing that like I didn't add a rename file, even though I had a rename flag selected. So it tells you here what's going wrong. And if not, if you don't get a good enough error message here, let me go at another here. So for example, is anything running that I can show? Oh, here. Okay, so this little page with the cloud icon is basically the backend log. So if you open that, it will show you a lot of things that's happening that are usually fine. It's just the VM putting out a lot of information out there. But if there's something wrong, this little icon here will turn red, and there'll be like two exclamation marks, such as, error, look here, kind of a thing. You can expand it, and it'll show you what's happening, and that would be the first place I would look if something's going wrong. Um, and yes, that's PRS and Anvil for you. There are some example visualizations that you can look at, and also the demo plot that's in there. You can use that to create this nifty plot. And I welcome you to use your own data, explore, and find new ways to create fun plots like this. Thank you.